are successful comparability studies designed and executed? And, what are those in process and release testing requirements? What does all of that look like for our audience?
So this is kind of a long answer. I actually have notes on this one. So a couple of points first, right? And this is something that, astonishingly to me, it's reared its ugly head over and over again. Comparability is not a voyage of discovery; it's a prospective activity. You have to ensure you have a pre-approved set of well thought out acceptance criteria in a protocol, pre-approved by your quality unit before you start. Okay, this ensures the application of the scientific method, really. And then also you have to ensure you have material available to evaluate a pre and a post change state. There was a question here that I got about the amount of retains that you need. This is an example of it. You may need more than the regulatory requirement for retains to be able to continually evaluate comparability as we evolve our product. The second point, we need to evaluate all the way down to drug product. It's not a drug substance issue, it's a drug product issue as well. So for example, comparability for a change in lentiviral vector, you should only evaluate lentiviral vector attributes, but forward process into DP to evaluate as well. So if you're changing your vector, don't just look at the vector process. Look at how the vector impacts your final drug product, okay? Ensure statistical confidence is achieved, right? I can't tell you how many times I've seen comparability exercise that we don't even mention statistics. Back in the early 90s, when I worked in this little product called Rituxan, we thought comparability was product A, process A meets specs and process B meets specs, we're done. Not necessarily true. In fact, it's not true. Okay, so the best comparability studies are really designed with a direct correlation to the product and process control strategy and risk assessments for both the product itself, that includes aseptic processing, okay, and a holistic view of the process. This is where I think I might disagree with John, where the product is the process. In this context, I think it is, because the aseptic processing, I don't care how you say the process is closed, it's not closed. You're taking apheresis or a tumor. That's an open process right there. The information must be linked to analytics so that a comprehensive and defendable study can be executed. So, in terms of process and release testing requirements, I think I'm expanding a little bit, but you have to have a well-defined preliminary process control strategy, review each unit operation and where the change is occurring for parameter and attribute relationships, and then select those attributes for comparability testing based on that body of knowledge. There's no cookie cutter answer. The QRM is supposed to inform this. And then the other point is, of course, there's testing that we know about regardless of the product. Safety testing, sterility, mycoplasma, endotoxin, product safety, could be potency, VSVG, et cetera, or product attributes, vector copy number, or percent of a particular cell type, or cell number, viability, things like that. But that's kind of what I think about when it comes to design and execution up through, testing requirements.
Yeah, good. Thank you. And John, I want to hand the floor over to you for the same exact question, because we did get this a lot in the outset. How are successful comparability studies designed and executed? And feel free to expand on what Mike just said too.
Sure. Yeah, I love the conversation, because I think, what we're seeing here is we've got two people, myself and Michael, who we know each other well. We agree on some of the most fundamental things in the industry, which really underlies a lot of the reason that we're having these conversations, right? Like, Michael and I probably talk about it offline, but some of the things we disagree on around processes, product, and whether or not allo is a better for donor-donor variability. These are conversations, hour-long conversations on themselves, but I think it's telling the state of the industry, because people with a lot of industry experience can't agree on some of these more fundamental things. To the question that was posed around design and execution. I actually, this is where I do agree a lot with what Michael has said. I think he gave you some great examples of the things you should be thinking about, and maybe distilling those down into one central premise, and that's around devising a strategy up front before you make changes to the process. What are your readouts going to be, and how do those truly measure comparability? Define the, if you can, based on some of the historical data, define the normal operating ranges of those affected unit ops in such a way that you can get to the finish line more quickly with creation or adherence to pre-existing CPPs. I think one of the things we again, a more higher level message is, comparability is not a single CQA. In fact, certain CQAs really are not tied to safety or efficacy at all, at least not without conjunction to other CQAs or release tests. I think we need to be thinking about the totality of the data, and I think if you go to the agency, that's a messaging or a theme that they try to get across to innovator companies and drug developers is, don't think of in terms of a single assay, but the totality of the data that you're creating. And again, that ties back to what I said earlier. And I think that the general theme of what Michael was trying to get across, and that is really developing your process control strategy in such a way that, on the back end, what good looks like. You're defining good from the very earliest stage. And then on the back end, you say, oh, here we are. We're either hitting the mark or we're missing the mark.